हॅलो फ्रेंड्स, राणा अकॅडमी या यूट्यूब चॅनलवरती आपल्या सर्वांचं स्वागत आहे. आपण स्टार्ट केली आहे महाराष्ट्र स्टेट बोर्डची 11th क्लासची बायो सिरीज, ज्याच्यामध्ये आपण आतापर्यंत पाहिला 6 चॅप्टरपर्यंत. त्याच्यानंतर आपण सुरुवात करणार आहोत 7th चॅप्टर सेल डिव्हिजन. 6 चॅप्टर होता बायोमॉलिक्युल्स, आता 7th चॅप्टर आहे सेल डिव्हिजन, महाराष्ट्र स्टेट बोर्डचा 11th क्लास बायोलॉजी सिरीज. तर चला तर मग व्हिडिओला सुरुवात करूया. तुम्ही या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला सबस्क्राईब करून ठेवा, जेणेकरून तुम्हाला आमचे नवीन नवीन व्हिडिओज पाहायला मिळतील. चला तर मग सुरुवात करूया. लेट्स स्टार्ट. Life of all multicellular organisms starts from single cell, that is zygote. Growth of every living organism depends on cell division, as stated in the cell theory. Every cell arises from the pre-existing cell cycle. Sequential events occurring in the life of a cell is called a cell cycle. There are two phases of cell cycle, as interphase and M phase. During interphase, cell undergoes growth or rest as per the need. During M phase, the cell undergoes division. Interphase and alternates with the period of division. Interphase is the stage between two successive cell divisions. It is the longest phase of cell cycle, during which the cell is highly active and prepares itself for cell division. The interphase is divided into three subphases, or as the G1 phase, S phase, and G2 phase. G1 phase. This is also known as first gap period or first growth period. It starts immediately after cell division. Cell performs RNA synthesis, mRNA, rRNA, and tRNA, protein synthesis, and synthesis of membranes during this phase. Yes phase. It is synthesis phase in which DNA is synthesized or replicated so that the amount of DNA per cell doubles. Histone proteins are also synthesized during this phase. G2 phase. G2 is the second growth phase, during which nucleus increases in volume. Metabolic activities essential for cell division occur during this phase. Various proteins necessary for cell division are synthesized during this phase. Besides, RNA synthesis also occur during this phase. In the animal cells, a daughter pair of centrioles appear near the pre-existing cells. M phase or period of divisions. M stands for mitosis and meiosis. M phase involves karyokinesis and cytokinesis. Karyokinesis is the Pyrokinesis is the division of nucleus into two daughter nuclei, whereas cytokinesis is the division of cytoplasm, resulting in two daughter cells. Types of cell division. Three kinds of cell division are found in animal cells. They are mitosis or direct division, mitosis or indirect division, and meiosis or reductional division. Mitosis can be performed by haploids as well as diploid cells, but meiosis can be performed by diploid cells only. In honey bee, drones develop from haploid fertilization eggs, whereas in Merchantia and haploid spores form a gametophyte by mitosis. Amitosis. It is the simplest mode of cell division. During amitosis, nucleus elongates and the constriction appears somewhere along its length. This constriction depends on and divides the nucleus into two daughter nuclei. This is followed by the division of the cytoplasm, which results in the formation of two daughter cells. This division occurs in unicellular organisms, abnormal cells, old cells, and in fetal membrane cells. Mitosis. This is the type of cell division in which a cell divides to form two identical daughter cells, which are identical to the parent cell. It is completed in the two steps, as karyokinesis and cytokinesis. Karyokinesis is the small nuclear division which is subdivided into prophase, metaphase, anaphase, and telophase. Also, for the sake of convenience, above mentioned steps are used. It must be remembered that mitosis is a continuous process that starts with the prophase and ends with separation of two fully formed cells after cytokinesis. Prophase. And this phase involves condensation of chromatin material, migration of centrioles, appearance of mitotic apparatus, and disappearance of nuclear membrane. Figure 7.2 prophase. Figure 7.2 prophase. Due to condensation, each chromosome becomes visible under light microscope, which can be seen in its sister chromatids connected by centromere. The nucleolus starts to disappear. The nuclear membrane disintegrates and disappears gradually. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Mitotic apparatus is almost completely formed metaphase. In this phase, chromosomes are completely condensed so that they appear very short. Sister chromatids and centromere become very prominent. Now, all the chromosomes lie at equatorial plane now of the cell. This is called a metaphase plate. Mitotic spindle is fully formed. Spindle fibers are attached to centromere. Centromere of each chromosome divides horizontally parallel to long axis and to two, each being associated with the chromatid. Figure 7.3 metaphase. Anaphase, and the chromatids of each chromosome they are separate and form two chromosomes called the daughter chromosomes. The formed two chromosomes are pulled up away in opposite direction by spindle apparatus. Chromosomes being pulled away appear like a bunch of banana during anaphase. Each of chromosomes each set of chromosomes reach at the opposite poles of the cells marks the ends of the anaphase. Figure 7.4 anaphase. Telophase. The telophase is the final stage of karyokinesis. The chromosomes with their centromeres at the poles begin to uncoil, lengthen, and lose their individuality. The nucleus begins to reappear. The nuclear membrane begins to appear around the chromosomes. Spindle fibers break down and get absorbed in the cytoplasm. Thus, two daughter nuclei are formed here in a cell. Figure 7.5 telophase. Cytokinesis. The division of the cytoplasm into two daughter cells is called cytokinesis. And the division starts with the constriction, generally at the equator. This constriction gradually depends on and ultimately joins in the center of dividing into two daughter cells. This process of division of cytoplasm is perpendicular to the spindle. This mechanism of cytokinesis is characteristic of animal cells. However, plant cells are covered by a relatively non-flexible cell wall. Due to this, furrow cannot be formed. Instead of a cell wall, partition starts to appear at the center of the cell and grows outwards to meet the extinction existing meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. Significance of mitosis. As mitosis is equational division, the chromosome number is maintained constant. It ensures equal distribution of the nuclear and the cytoplasmic content between the daughter cells, both quantitatively and qualitatively. The hereditary material DNA is also equally distributed. It helps in the growth and development of organisms. Figure 7.6 cytokinesis. Old and worn out cells are replaced through mitosis. It helps in the asexual reproduction of organisms and vegetative propagation in plants. The process of mitosis also maintains the nucleocytoplasmic ratio. Mitosis is a very reliable process for preserving the genetic makeup of cells and organisms, but it cannot introduce variation or new combination of existing genes. Death of cell, you may think of it as a bad or bad for cells in your body to die. In many cases that's true. It's not good for cells to die because of an injury, for example due to scrape or harmful chemical, which is called necrosis. However, some cells of our body die not randomly but in a carefully controlled way. For example, during the embryonic development, the cells between the embryonic fingers die in the natural processes called apoptosis to give a definite shape of the fingers. This is a common form of programmed cell death where cells undergoes a unicellular cellular side. Now, when they receive certain signals, apoptosis involves the cell death, but it benefits the organism as a whole. For instance, by letting fingers develop or by eliminating potential cancer cells. Meiosis. The term meiosis was coined by J.B. Farmer in 1905. It takes place only in reproductive cells during the formation of gametes. By this division, the number of chromosomes is reduced to half. Hence, it is also called a reductional division. The cells in which meiosis takes place are termed as myocytes. Meiosis produces four haploid daughter cells to form a diploid parent cell. Meiosis is of two subtypes. First, meiotic division or heterotypic division, meiosis 1. Second, meiotic division or homotypic division, meiosis 2. First, meiotic division or heterotypic division. During first meiotic division, the diploid cell is divided into two haploid cells. The chromosome number is reduced to half of the total number of daughter cells. Hence, this division is also called a heterotypic division. It consists of the phases like prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. Prophase 1. This phase has longer duration, significant figures which are peculiar to meiosis occurs in this phase. This phase can be subdivided into five substages as leptotin, zygotin, pakitin, diplotin, and dikinesis. Leptotin. The volume of the nucleus increases. The chromosomes become distinct, long thread-like and coiled. They take up the significant orientation and the bucket stage inside the nucleus. This is characterized with the ends of the chromosomes converged towards that side of the nucleus where the centrosome lies. The centrosome duplicate the migrate to opposite poles. Zygotin. Intimate pairing of homologous chromosomes takes place by formation of a synaptonemal complex. This pairing is called a synapsis. Each pair consists of a material chromosome and a parental chromosome. Pakitin. Each individual homolog chromosome has two similar chromatids. At this stage, tetrads become more clear in appearance because of presence of four visible chromatids. The homologous chromosomes of each pair begin to separate from each other. However, they do not completely separate but remain attached together at one or more points. These points appear like a cross known as chiasmata. Chromatids break at these points and break up broken segments are wrongly exchanged between non-sister chromatids of homologous chromosomes. This is called as crossing over or recombination. Diplotin. Through the chiasmata are formed in pachytene, they become clearly visible in diplotin due to the beginning of repulsion between synapse homologous chromosomes. This is called as dysynapsis. It involves disappearance of synaptonormal complex. Metaphase 1, the spindle fibers become well developed. The tetrads move towards the equator and they orient themselves on the equator in such a way that chromosomes centromeres of homologous tetrads lie towards the poles and arms towards the equator due to increasing repulsive forces between homologous chromosomes. They are ready to separate from each other. Figure 7.8 Metaphase 1 Anaphase 1. In this phase, homologous chromosomes are pulled away from each other and carried towards the opposite poles by spindle apparatus. This is a disjunction. The two sister chromatids of each chromosome do not separate in meiosis 1 dikinesis. In this phase, the chiasmata begins to move along the length of the chromosomes from the centromere towards the ends of the chromosomes. The displacement of chiasmata is termed as determinalization. The terminal chiasmata exists till the metaphase, the nucleolus disappears, and the nuclear membrane also begins to disappear. Spindle fibers starts to appear in the cytoplasm. Lactotin, zygotin, pachytene, diplotin, dikinesis. Figure 7.1. This is reductional division. The sister chromatids of each chromosome are connected by a common centromere. Both sister chromatids of each chromosome are now different in terms of the genetic content, as one of them has undergone the recombination. Figure 7.9 Anaphase 1. Telophase 1. The haploid numbers of chromosomes are after reaching their respective poles now become uncoiled and elongated. The nuclear membrane and the nucleolus reappear, and thus the two daughter cell nuclei are formed. चला तर मग आपण हा व्हिडिओ इथंच थांबूया. नेक्स्ट व्हिडिओमध्ये आपण पाहूया. चला तर थँक्यू सो मच फॉर वॉचिंग. प्लीज लाईक शेअर अँड सबस्क्राईब.